Hello, hello. Welcome back to the Baby Blueprint. I'm Sarthak Sawarkar here with Santiago Munné. As you folks know, the show started off from the conversations Santi and I were having every day. The conversations were about fertility, genetics, embryo science, and generally where human reproduction is headed. We are not professional podcasters. We are not media trained. We're just trying to have curious, evidence-based, and hopefully useful conversations with people who are shaping the field. Today, we are joined by Nuno Costa-Borges to talk about the future of embryology, IVF science, and how the lab itself is evolving as reproductive medicine becomes more precise, data-driven, and accessible. Me and Santi had an argument at one point in time, not that far back, about whether the role of humans in the lab, in the IVF lab, generally was over. I said no. Santi definitely seems to be on the side of yes. So I think we're going to have a great conversation today. Santi, I'll hand it over to you to introduce Nuno.
Sure. Thank you, Sarthak. Yeah, it's a great pleasure to introduce Nuno Costa-Borges, a good friend. We have collaborated in the past. He's actually the founder and CEO of Embryo Tools since 2013. And also is the director of experimental biology at Conceivable. Basically, his claim of fame, I would say three. It will be maternal spindle transfer, right? He has the first babies doing maternal spindle transfer, automation of ICSI. We have together the first babies after automation of ICSI, and then basically back to back the whole from the start to the end of the process of ICSI with Chavez Badiola. So he's an amazing skilled embryologist. I mean, I've seen him working with his hands with the macro manipulator. And obviously the first question that I have is, well, if we automate, are you going to miss it?
Yeah. Well, that goes with the assumption that we are going to automate, which I don't think so. So before we go ahead, I mean, Nuno, welcome, man. Really appreciate you joining us.
Thank you so much to both of you for seeing on me to participate in this podcast. And it's a pleasure, of course. And thank you, Santi, for the kind introduction and words. Coming from you, it's an honor.
And Nuno, so Santi, I ended up spending time with Nuno, his wife, randomly one of the days when I was visiting Spain, and they're one of the nicest and kindest people that I've spent my time with. I still tell everybody, they're really, really nice folks. Well, I mean, we could start with Santi's question, right? Like, what led to this? How did you get into the field? For any aspiring embryologists, maybe a quick sort of a background in terms of how you ended up here.
Yeah, well, I started in 2003 as a PhD student in a project that aimed to increase the efficiency of somatic cell nuclear transfer, cloning in animals. So I did my PhD there at the University Autónoma of Barcelona as a fellow of a Portuguese foundation for technology, something called like that, from the government. I had a grant, and I had the chance of working with a micro manipulator since I was a child, more or less. But, more than 20 years ago. And I remember by that time, everything related to biotechnology and reproduction was a kind of a boom, because there was the dolly had been cloned. There were several animals that had been for the first time shown that could be cloned. There was also an interest in doing a somatic cell nuclear transfer for therapeutic purposes to obtain stem cells that could be a source for curing diseases. So there was, by that time, there was a great interest in all these type of technologies. So I had a chance of working in this project, and then this opened me the possibility to work as a clinical embryologist in a clinic in Barcelona, for EV group, and in there I had the experience of being a clinical embryologist, so I had already the background of being a researcher, and both combined, the clinical part and the research part, gave me this background that we have now, that allows us to do research, apply the research to see if we can improve certain parts of the clinical work that we believe there's still some space for improvement. Yeah, so that's where we stand now. Right?
And do you think that right now, well, I have at least, and a lot of people have said the same, that most of the embryologists right now are basically following protocols that have been developed for a long time. And some of them, or I wouldn't say most of them, let's say most of them are just following protocol instead of developing protocol. What's your impression of the new embryologist?
Well, I think since because our field, as everyone knows, it's relatively new, and for a certain period of time, there was this interest in developing new technologies, all that came in the 90s and beginning of the 2000s, all interest in improving, but then we reached, I think, a plateau where novel technologies didn't demonstrate to be a really breakthrough for the field. So in the end, and also as part of the clinical work, you cannot change protocols that have been implemented. So it's part of the clinical routine. Otherwise, it will be very difficult to control the lab. So in a sense, I think clinical embryologists do what they have to do, that is follow protocols that have been already established and are good to overcome certain infertility problems. But it's true that on the other hand, if there is no progress, no improvements, we will not be able to tackle problems that today with conventional treatments we cannot solve the problem, right? So we reach this paradox of how to push the boundaries of the field, but also embryologists cannot deviate from what is standard and what is considered the routine work. So yeah, in the end, the more they have to do and the more demand for conventional treatments, the less they can be used, or they can use their experience to improve new things or to develop new protocols. So we are in this paradox. Yeah.
Yeah. So probably with the private equity buying a bunch of clinics, standardizing even more everything to make it more efficient economically, that also is contributing to not a stagnation, but yeah.
Why would you call it stagnation? I mean, in my opinion, I think the standardization is probably contributing towards better results, right? Like what kind of improvement would you be looking at this stage? I mean, help me understand this. I think there's a little bit of a background that maybe we can provide the folks that are listening in, especially those that are not directly VCs. Those are not directly in the field of IVF or fertility, but are maybe joining us as VCs. They're joining us as folks from the outset, right? They're looking into the field that looks like a black box to them. They know that there is, they loosely understand what IVF is, but as an embryologist, right? What do you do, right? Like, what is it that you do? Are you, a doctor, or what is it that you do? I know that. I just want to make sure that we make it clear to the folks that are listening into us.
You mean in general embryologists or myself? Or
generally, what would embryology entail, right? Like, is it a medical science? How would you qualify it? What is the degree that somebody has to pursue to become one? I know what you did. You just told us that.
Yeah.
But what is it? What does a day-to-day look like maybe for an embryologist?
Yeah, typically embryologists have a background in science, in biological sciences, and some of them have more specialization, doing masters or even PhDs. But in the end, what really is needed is good hands. And as Santi was explaining, there's not too much space for deviating from the protocols that are already established in the clinic. And because everything is very manual, in order to be proficient in doing these protocols, they need a lot of training and practice, and then there's not space for improving or for developing other things that maybe they're missing. In a daily routine, you have all the procedures that are needed from this preparation, which is a very tedious but very important task of an IVF cycle. Then sperm preparation, oocyte retrieval, oocyte insemination or ICSI, then embryo culture, embryo biopsy eventually, oocyte and embryo vitrification. So typically, these are the steps that are needed to complete an IVF cycle. And whenever it's possible, and I guess in most of the labs have this well implemented, you have to have a good control of all documents, a good traceability of all disposables, plasticware, labware that is used in the lab, time to ensure that the equipment is working properly. So you need to have time to do quality control, quality assurance in the lab. You have to organize to have a good access of the data and analyze them periodically to ensure that the results that are obtained by the lab and within the different individuals, operators that work there, that follow the minimum or that reach the minimum competency values. So it's a lot of work, and as Santi was explaining before, now with the acquisitions and the demand for IVF treatments, I mean, the persons in charge want the lab to be the most efficient possible in terms of performing the cycles and the number of cycles, and there's not much space for doing research or for trying to improve what is already established. It's true. So in the end, embryologists become most of them followers of protocols, or in other words, technicians in embryology. Yeah.
So then, Santi, where are you expecting that innovation to be built in by an average embryologist? I mean, the question sort of was something that led me to understand that, there is some possibility, but Nuno is making it out to be a very laboratory sort of science that's guided by clinical protocols, etc. Where do you think we can?
Well, yes, there are two things, right? One is, okay, if we are following protocol and it's always the same, then it can be automated, right? And this is what's going on right now. On the other side, we still have, even when you do PGT, when you select the embryos that they don't have any chromosome normalities, you have implantation rates that top a maximum 70%. So we still have 30% of the eggs, embryos that don't implant, and we don't know why. So still there is a lot of room for improvement.
Yeah, I agree. And I think everything is converging towards the same direction, right? And I think most of us agree that certain parts of the lab can be automated. It doesn't make sense to have someone dedicated to do things that can be even done or performed more efficiently by an automated system, right? But before reaching that point, I think the trend in our lab currently, and probably it started without us noticing some years ago, it's the simplification of procedures. We are assisting to a simplification of protocols of procedures to make them more simple for an operator, and so that they can be more consistent as well. We have different examples. So we started with embryo culture, If Santi probably remembers the time of Life Global, where most people couldn't believe that embryos could be cultured in a single step using a unique solution.
Mhm.
Then with the integration of this simplified a lot what is the culture system, because before we had to move the embryos from one dish to another in the middle of the 5 or 6 days of culture. Then the time-lapse systems came, which was a perfect fit for the single-step medium, for one-step culture. Now we are insisting that, for example, sperm preparation can be done with just doing a microfluidic chamber. So there's no need for centrifugation and no handling that was needed. We're seeing vitrification. That was also a protocol that involved several steps, and how there are more and more evidence that the warming of the embryos can be done just also in one step. We have a paper in review that has been submitted, that is in revision now, that showed that also vitrification can be done in one step. So if all the trend is to simplify things. So it's not, before, when all these procedures were developed, it was thought that we needed to have very complex with different steps and so on. And now we're seeing that probably we can simplify all this. And if we simplify this, we're opening doors for automation, because it's easier for a system to pick one embryo, move it to one droplet, and from that droplet to vitrify it, to do insemination, whatever. And if the handling is complex, I mean, little by little, everything will converge to what will be the simplification of the lab, introduction of automated systems. And with this, probably, I think I'm not afraid as an embryologist, and it's something that I tend to explain everyone, because we'll be lucky of assisting all this wave of automation entering in the lab. And on the other side, we'll have much more time to develop things, as Santi was explaining, trying to overcome situations, extreme or more extreme cases of infertility that for now we don't have a solution, and can be very frustrating for patients and for doctors as well, because they feel that patients repeat cycles and cannot be offered something really tailored for their needs. So I think this will be the future, and we'll be lucky that.
Well, I mean, I think so. Here is where I think I'm diverging a little bit, right? Which is the fact that there is a totally inordinate amount of attribution, I think, something from your perspective, that 30% gap in implantation failure is because of the lab, right? I don't know.
No. I'm not saying it's the lab. I'm saying that we don't know where it is. And
Sure.
that's in the top IVF lab, right? So with automation, we can assume that we'll get to the 70%. Now, that was the next question I had for Nuno. And it's right now, I mean, basically, well, I work with Overture. He also worked with Overture in the past, now Conceivable. Both companies, and I guess the others also that are trying to automate IVF, basically are mimicking the manual processes, right? To get to the same level as manual. The thing is that basically we're teaching these devices to follow a protocol. So this device cannot think about how to improve this protocol. So yeah, that's again, it could be another limitation that we're self-imposing in the field, because these machines right now are not designed to improve the protocol by themselves.
Well, let's ask Nuno the question then, which is like, are you employing the new wave of AI into understanding how these limitations can be solved, and how are you using them? What is your thought process? And not specifically, but fundamentally, what is where do you see AI integrating into the process?
I mean, AI was something that was missing in the puzzle, and since a few years back, we had the opportunity to introduce it so that the systems that involve automation can be more autonomous. So they can be consistent and autonomous. Autonomous in automation is very important because it means that it reduces the level of intervention of a human being. And for now, or some years ago, when there was attempts to automate things, I mean, the robotics part was well developed, even microfluidics was well developed, but we are missing how to instruct these robots to do the tasks. And now with AI, one of the applications, I mean, most in our field has been used for selection of oocytes and or embryos for transfer, right? But I think where AI will play a very important role is also in automation to provide instructions for the automatic systems on how to execute the different tasks. And this where I believe it was the piece that was missing for automation. But currently it's well developed and it will be very helpful for all this.
Yeah. But so yeah, you're solving the how you call it, the autonomy of the system. But can the system
be better than embryologists? You mean?
Yeah.
No, I guess I think we're at early stages of automation, right? So the aim for now is to be at least, or to provide more consistency for the different procedures that are done currently by manually by the operators. And so this is the first goal. The first aim at these early stages of automation, I think for all companies that are working in automation, Conceivable, Overture, Auto IVF, there are several. And the first goal is to at least be as good and more consistent as a human being. And once all this will be well established, and I believe it will take some years from now, then we'll be more accurate in doing diagnosis for patients, because we'll know that there will not be the fluctuations that we're seeing now depending on these patients are treated. So we'll assume that all labs will all have similar consistency. So when something fails, probably it's because of a biological nature, not because of a technical problem or something that was not a suboptimal condition of the embryo culture, something like that. And that's where then we'll be able to tailor the treatments for patients, for doing personalized treatments, because probably the automated labs can solve the majority of the infertility problems or subfertility problems, but all those patients that are more complex cases probably won't be able to be treated with automated lab, and they will need tailored specific techniques for their problem. And there's where I think embryologists can also play a role, because they will have more time also to develop new technologies that currently we're lacking. I'm thinking about, as you mentioned, Santi, implantation failure, patients that don't have enough, or they lack at all. They lack gametes. For example, there's a lot of work being done in gametogenesis, for example. Other are focusing in more advanced stage patients, trying to avoid chromosomal errors, so doing treatments tailored for oocytes of advanced stage patients, patients with mitochondrial diseases, all these sort of treatments that currently are still experimental, and if we don't push them, they will be still in the closet.
That's a great vision. and I totally coincide with you. So let's change, unless you, Sarthak, has other questions about the, automation. Let's change subjects and let's talk about how to treat these special patients that have more attention, like repeated IVF failure. So I had a question about this, the maternal spindle transfer. Do you think that you can fix aneuploidy with it, or it's too late?
Yeah, that's a good point. So with the evidence that we've been able to collect for now, I think we're certain that if we put a spindle coming from an outside that has been exposed to oxidative stress, that has cytoplasmic dysfunctions like mitochondria or maternal transcripts, and you put it at metaphase 2 stage in another healthier cytoplasm, we are quite convinced that, and the evidence shows that we are able to increase or overcome embryo development arrest. So this spindle in a new engine, in a new cytoplasmic machinery, can make it more blastocysts. But as you mentioned, at metaphase 2 stage, we need this spindle to be euploid. We need to ensure that the first meiotic division is done correctly, so that when we put it in the new cytoplasmic content, we can give support for the spindle to give rise to an embryo that is chromosomally normal. So for advanced stage patients, probably it could be a good treatment if within their cohort of oocytes there is still a few that are chromosomally normal, because we would ensure that with those ones, when we transfer to a new fresh cytoplasm with a healthier mitochondria and other organelles, that we have more chances of having blastocysts. Mm-hmm. We would end up doing biopsies in more blastocysts, but we still need to have one or two oocytes that was euploid before the cycle. So,
do you think that these patients that have repeated IVF failure, obviously, yeah, the ones that you choose for this treatment and it work, what was the reason that they failed? Do you think it's mitochondrial dysfunction? It cannot be genetic, right? Because if you put it in another cytoplasm, then it works.
There's some interesting cases of patients that have variants in their exome that showed that with these variants there are some maternal transcripts that are lacking in the oocytes, and these are important for the first stages of development, where all maternal transcripts are essential to support the embryo development. So when transferring this spindle to a cytoplasm that contains these proteins that are lacking, we have shown in animal models that this can be also a solution for the problem. And because it's very difficult to identify what is failing in a cytoplasm, because it's composed by thousands of mitochondria, thousands of maternal transcripts, thousands of organelles, and it's really difficult to identify what's the real cause. But what the truth is that there's some specific patients that are still not too old, that when they repeat the cycle several times, the same phenotype of embryo development arrest persists, and most of them end up of being or giving up the treatment or being offered oocyte donation, which for them can be very frustrating, because most of them are still young, and it's not because they delayed the wish of being mothers, right?
Interesting. I was reading the other day a paper on gene editing, and when they transfer RNA as the driver, then the eggs basically stop developing. So interesting. That could be one of the reasons, right? Some RNAs that are either not there or
are defective. Yeah.
That's the most likely explanation, as you would find it anyway. Is there a graphic that you can point us to, Nuno, while you were talking about this? Just because, again, like nuclear spindle transfer is so cutting edge that I don't think any of our listeners would have ever heard about anything like this, unless they're very deep within the fertility field. Do you have a graphic that you can pinpoint us to that you can walk us through on this one?
Essentially, these are cases where oocytes stop their development, or they either fail to be fertilized, or they stop their development. They arrest the development within the first days of in vitro culture. So typically, it's within day 2, day 3. Most of these oocytes don't develop further, so they cannot reach the blastocyst stage. So by transferring the meiotic spindle, that's where the genomic DNA is present, to a cytoplasm from a donor, to an oocyte from a donor, from which we removed previously the genomic DNA. We are providing this spindle a new cytoplasmic content, a new environment. Assuming that comes from a donor, it will be healthier than what was previously or originally present in these patients that repeated the cycle several times without success. So we've seen that patients that pass through on average of 6.6 previous IVF attempts with the same problem persisting due to embryo development arrest, using only an average of 4.4 oocytes per case, we were able to get 60% blastocyst, good quality blastocyst formation rates. So it was very, it was a pilot trial. Trials per definition don't aim to look at efficiency, but it was very clear that something can be, this pattern can be overcome with this technology.
That's amazing. So what I understand it as is that once you actually make the embryo, what you're doing is in the very early stages, you're moving the nucleus from one from that into a totally new egg from a donor, in which case this is a totally unrelated female donor that is donating their eggs. And the machinery, the workshop is theirs, but there's no genetic similarity with that, other than I believe the mitochondrial DNA from the donor, which would be the only thing that is different. And you're saying there's extraordinarily high quality blastocysts, right?
Yeah. Sorry, Sarthak, you define it very well, and it's for giving the patients the opportunity of having a child genetically related to them. So rather than,
And then, have you actually implanted any of these embryos, and do we have any kind of outcome data that?
Yes, have babies.
Yeah. There we have 7 babies that have been followed up, and now we're planning to publish a follow-up paper because they're now around 5 years of age. As another evidence about the safety of the technology, because this is something also important.
And are you doing any kind of mitochondrial sequencing on the donor before you take them on, just to understand that there's nothing genetically deficient in the donor?
Yeah, I mean, we try to use haplotypes that are similar to the patient. So there's a few variants in the mitochondrial DNA sequence that differ between the patient and the donor. In this particular study, the greatest difference between patient mitochondrial DNA and donor was only 0.2%. So it was haplotypes quite similar between patients and donors to ensure also there's no incompatibility between the nucleus, the nuclear DNA, and the mitochondrial DNA. Yeah.
Fascinating. Simply fascinating.
Uh,
Yeah.
I was going to ask about regulation and all the hoops that we have to go through right now to do all these experiments, and it's getting more and more difficult to. Do you think that we are beginning to be over-regulated to European?
Well, I think it's important that everything that is new is well evaluated, that we pass we follow all the steps to ensure that when we do the translational work to humans, we don't find we don't have any surprises. And but of course, at the same time, while we're developing all these, I mean, the regulators need also to be aware of everything that is emerging in the field. We cannot be waiting for them to evaluate something that is already proven to be effective or safe, and wait many years to get these approvals. Because what happens is most of the promoters or developers of these technologies are not waiting for all this long. They need to find alternatives, and maybe they go to less regulated places where probably the follow up or the regulatory is less strict, and maybe we lose control of what's going on in those countries. So it would be important in order for I mean, it's a call for transparency as well, no, and also to provide patients the high standards possible, that the regulators do their job and they're up to date to what's going on in the field. And whenever emerging technologies show to have passed through all the research steps and all the evidence showed that the technology is ready to be used clinically, that they do also the step of regulating it.
I mean, I totally agree with you, right? I mean, one of the things I was just thinking about as you were talking about this is most people who freeze eggs never end up using them, right? This technology effectively lets these folks donate their eggs without being worried that they'll have genetically related children running around that they're unaware of. So as long as you're following, like regulators are on speed, there's, in the US, you have FDAs, etc., that you are doing, plus you have sequencing probably being done ahead of time, right? At full maturity, every single egg that is in storage can potentially be just used as a new house for folks that are trying to figure out unexplained infertility, or I don't know what exactly you're calling this treatment for, but wow, just it's totally going to be it's gonna need thousands of more embryologists, Santi, because this is definitely not going to get automated anytime soon, right?
That's going to be a difficult one to do.
It is going to be, nothing's impossible, but yeah. This all started with one of my cousins who just graduated college, who loves playing video games, and I was telling him he should become an embryologist, and me and Santi basically went back and forth on whether embryology would exist or not. But I think you've made a case that, from Santi's perspective, what he was saying was accurate, that a lot of bread and butter embryology as we know it might get automated sooner than later, but then there's so much more that can be done with the science that we should definitely keep doing it. Very cool. I think we can, Santi, unless there's anything extra, I thought we could have a quick rapid fire round with Nuno about just a few questions, and you can feel free to say pass if you don't feel like answering them. But I thought it would make it a little bit more fun and interactive.
Be so difficult?
Oh, well, I don't know. It may be political, but embryology in one word: art, science, or engineering?
Science.
What is the most overrated metric in the IVF lab?
You're talking with the wrong guy. For me, everything is important.
Everything is important. There you go. Well, what is the most underrated thing? What is the thing that people don't pay enough attention to that affects embryo quality?
I think the air quality still plays.
Air quality?
Yeah. Mm-hmm.
Fair enough. I think we've had this conversation in the past. I could see that. If you could remove one bad habit from every IVF lab tomorrow, magic stick, what would it be?
Oh, that is difficult. A bad habit. Doing ICSI with aspiration to break the membrane. Yeah.
All right. I have two more questions. Yes, sorry, Santi, go ahead.
No. I was going to ask, do you think that the doctor will be automated? Because we're talking about only automating the embryologist, right? But
yeah, that. Then we should go to the previous question, right?
Well, moving on, what is the one thing from the patient's perspective that you would want patients to understand that embryologists bring to the table? Is there something that you want, if a patient's listening to this directly, generally the patient's only interaction point is the doctor, which according to Santi could be automated soon. In my opinion, that's not something that is, at least the management side, I think we would see a lot more automation around. But from an embryologist perspective, what do you think a patient should realize that you control? Or don't control, right? Like both ways. Like
I mean, that in the end, embryologists work with the biological material that they get. So there's sometimes that is
right.
able to improve what the aim is not to reduce competence, but things to improve it's difficult to improve what we get, right?
That's such a good point. I mean, I think it is, there's two sides of the same coin here that you're talking about. So I'm going to take the liberty of just adding the other side to it. One is that it is, one thing that they should understand, that it is the biology that governs most of the outcomes, is what you're saying here.
Yeah. Mm-hmm.
The second thing, though, I think is recognizing that while this is still done by humans, all humans are not the same. Mm-hmm. And outcomes are going to change between embryologists and between, cycles effectively, right? Mm-hmm. So because having run a clinical startup now for the last 4 years, I've heard all versions of, but why was this cycle like this? Why were outcomes like this? So this is such a great point.
Yeah.
But I don't have anything else for the time being. This was exceptionally fun and so enlightening. Santi, Munné, closing remarks from you, and then we can call it a wrap.
No, yeah, that was very interesting. I think we agreed that although how embryology right now can be automated to a certain point, like this maximum of 70% implantation, we still need great embryologists like Nuno to get to 100%.
I love that. And that's pretty much it. Thank you so much for joining us, folks. This is the Baby Blueprint. I'm Sarthak Sawarkar. That's Santiago Munné. Thank you so much, and see you in the next episode.